that laboratory in 1980. In 1981, he received the Arthur S. Fleming Award for Outstanding Government Service. And in 1987, the year of his untimely death at age 43, he was elected to the National Academy of Sciences. You can tell I'm a Mac person. And this is a photo of George Khoury on your left with a very young Peter Howley and Malcolm Martin, who was my own PhD mentor, or postdoctoral mentor, and is here in the audience. So during his short career, George published over 140 papers deciphering mechanisms of eukaryotic transcription. Many of these use viruses, and in particular SV40, as model systems. And we have a Mac to PC garbling there. George had very distinguished trainees and collaborators listed here. And similarly, the George Khoury Memorial Lectures have been given by a very distinguished group of scientists, most recently Julie Pfeiffer, Carla Kierkegaard, Juan Chang, Britt Glassinger, and Susan Weiss. And this brings us to today's speaker, Ileana Cristea. She did her undergraduate and PhD work at University of Manchester in the UK in medicinal chemistry, proteomics, and toxicology. Did a postdoc at Rockefeller University before being recruited to Princeton University, where she rose really remarkably quickly through the ranks to become full professor in 2016. Ileana has received numerous awards, too many to list here, but these include an Avant-Garde Director Pioneer Award, Mallinckrodt Scholar Award, Discovery Award in Proteomic Sciences. She was director of the Graduate Studies program at Princeton, Graduate Mentoring Award at Princeton, and as of last year, editor-in-chief of Molecular Cellular Proteomics. Ileana has made really major contributions to several areas of research, including developing proteomic methods for studying virus-host interactions, organelle remodeling, nuclear sensing of viral DNA, sirtuins as evolutionarily conserved antiviral factors, and regulation of protein acetylation. So I'd like to welcome Ileana to the podium for her lecture, The Interface Between Metabolism and Immunity Within a Virus Microenvironment. Welcome.
Thank you so much for that very kind introduction. It's just such a great honor being here and seeing old friends and new friends, and just really, I cannot tell you how honored I am to be invited to give Dr. George Khoury Memorial Lecture. And I want to say a few words about this and why it means a lot. And I think I will say that Eric already touched on this in terms of fundamental scientific discoveries that George has contributed to the field of virology and just broadly to molecular biology. His work on enhancers and also on understanding transcriptional control of gene expression has been extraordinary. But I want to say also from the perspective of Princeton, Eric told you that George actually had his undergraduate degree at Princeton, and I just want to say that he became such a strong supporter of Princeton University and so committed to recognizing people who have contributed to training him. And he has been in constant kind of touch with Princeton University, becoming an advisor to the Princeton University president and really helping tremendously to create the Department of Molecular Biology and to recruit a number of faculty as kind of the inaugural faculty to start this new Department of Molecular Biology for Princeton. Amongst these were Arnie Levine, but also Tom Schenk. And Tom Schenk, a remarkable virologist, herpes virologist, who I got to collaborate with when I was at Rockefeller University as a postdoc. And he really encouraged me to apply to Princeton for a faculty position. And I want to say that George has impacted my career in another way as well, even earlier, in 1970s, when here at the NIH, he interacted with Dr. Lynn Enquist. So Lynn Enquist at the time was in a laboratory close by to George's laboratory here. And Lynn credits George for encouraging him and suggesting that he maybe should change, use his skills to changed some of his scientific focus in a courageous way from his work on bacteriophages and lambda-phages to eukaryotic viruses. And Lynn Enquist later on joined Princeton University, becoming the chair of the Department of Molecular Biology and being the one who actually ended up recruiting me and hiring me at Princeton. So I may not be here speaking to you without George's contribution to molecular biology and to Princeton. So thank you so much for having me here. A little bit about my talk today. As you heard, I would love to tell you about kind of how some research directions in my lab have evolved to look into at this interface between regulation of metabolism and impact on immune signaling in this context of virus microenvironment. And first I will say what my disclosures are. I'm a co-founder and member of this scientific advisory board for Everest Bio that focuses on antiviral treatment targeting sirtuins. And I'm also a prior member until recently of the scientific advisory board for 908 devices that are focusing on methods for separating proteins and for analysis with mass spectrometry. But what I will tell you is not connected in any way or shape or form with these two positions that I have held or hold. Okay, so the story that I want to tell you again, and to put it in context, started from our interest in understanding different types of cellular communication during viral infection. And of course, there are many different types of communication during infection in a host cell. So at the intracellular level, we all know that an infection triggers profound changes in a cell. So changes in not only the composition of a cell, but the organization of a cell, where organelles can be positioned differently and they can look differently. And I would want to spend the first part of my talk looking at how we looked at intracellular communication that is facilitated by organization changes and by changes in organelle-organelle contacts and what this means. And then I will transition to considering this infected cell as part of its environment, as part of its system, and looking at how this infected cell conveys this information and how it affects its environment, the cells that are in the immediate proximity or those that are further away. And now we have tried to kind of characterize this still not sufficiently well-charted environment of the virus microenvironment. And I'll try, as I look at these intracellular and intercellular stories that I will tell you about, what their relevance is in terms of infection and co-infections and disease and infection-induced pathologies. So to start at the intracellular level, as I told you, infection with viruses induce such profound changes in the organization of the cell. And a still underexplored area of virology is that of organelle remodeling. And that is so fundamental to any sort of virus that you can think of, that viruses need to, let me see if this works. I'm trying to, okay, I'm looking if I have a pointer. Maybe I'll just use this. Can you see my pointer? No. Okay. I'm getting very quick technical support, I see. No, it doesn't work. It's all right. I see a pointer on your presentation that appeared, so somebody was trying to help you to point it. Okay, so viruses induce these numerous changes in organelles, in organelle shape and composition, and that can be put under the umbrella of organelle remodeling. And these organelle remodeling events are so important for entry of a virus into host cells, for the replication of that virus, either if it's nuclear or cytoplasmic, for the assembly of new infectious viral particles, or for the egress. And I'm showing this here for beta herpes virus, human cytomegalovirus. It's really an important virus, widely spread and connected to lots of different chronic diseases, including cardiac disease, but also a leading cause of virus-induced birth defects. But we can look at many other viruses, and we will find similarly different range of organelle remodeling events. So, for example, HCMV, that I told you earlier, induces these changes in nuclear periphery and the assembly complex that you see on the right-hand side. HSV1 also targets the nuclear periphery. It also fragments mitochondria. Influenza A also remodels mitochondria. coronaviruses are inducing these double membrane vesicles formed of ER that are really important for the replication of this virus. So there are all these changes in the shape and the morphology of organelles that are clearly controlling and regulating the organelle function that happen during infection that are needed for infection to progress. But this, as I said, is still a kind of an underexplored area of virology. So for many of these events, we do not understand exactly what their functions are, or if they are connected in any way to some of the linked infection-induced pathologies. Also, how are these coordinated? Of course, if a shape is different, it's going to affect differently its immediate environment. Like if I spread my arms or if I'm all together, I'm going to impact differently and make different connections with my immediate environment in the cell. In the same time, how are they coordinated in space and time, or functionally coordinated? So we have investigated this for a number of years in my lab, and I want to give one example from some studies that we have done some years ago that really have started to push us more and more towards this idea of coordination, that these organelle remodeling events are actually linked to each other and they're affecting each other. So this was from work that we have done on peroxisome, where when we were studying human cytomegalovirus, and we have done this also with HSV1, we started to separate organelles by density fractionation, characterize their composition, and find out how do they change. And then we combined this with structural microscopy studies and also with lipidomic studies and mathematical modeling and machine learning to start to learn and assign components, viral host components, to different organelles, or start to understand what does it mean in terms of these structure-function relationships. And what we found is that peroxisomes, I'm still hoping that I can get the pointer here. Let me see if I can do this. Perhaps.
It's not working?
No, it's not working, but maybe I need to, yes. Yeah, it's all right. I'm just going to, so in this circle here, you can see peroxisomes. So we found that peroxisomes are changing in shape during the infection and also in the number, and that this change in morphology is activating enzymes, peroxisomal enzymes, that are responsible for initiating lipid synthesis, the synthesis of lipids called plasmalogens. And we showed how these plasmalogens are really critical for the secondary development of HCMV and becoming components of infectious viral particles. So the reason why I'm saying coordination is because this lipid synthesis starts in the peroxisome, but it's actually the lipids have to be matured in the ER. So this synthesis is based on the interaction of peroxisome with the ER and the formation of ER peroxisome contacts. So the more we interrogated organelle remodeling events, the more we were struck by how interconnected these changes are and based on contacts. And we all know that organelles do not function in separation, but rather they form these intricate networks of communication inside the cell that are so important for so many fundamental cellular processes. And these interactions are facilitated by proteins called membrane contact sites. I see some promise. No?
Option one, you
Yes.
have to activate it on here.
All right.
It might be this little button.
Okay. Ooh.
Oh, I hear you.
No worries, no worries.
Okay. And then,
Victory.
okay.
Yes,
Otherwise,
okay.
it was me pointing.
That I would have loved to see that, Chris. Yes, I would opt for that second option. Okay. So, all right. So these interactions between organelles are facilitated by these proteins called membrane contact sites that are bringing organelles in close proximity. So these are not fusion events. And they can facilitate direct transfer of molecules like lipids and calcium. And I'm showing you here one of our microscopy images where you see in red peroxisomes and in cyan ER, and you see how dynamic this interaction is. And I was just telling you on the previous slide about this ER peroxisome contact. So we wondered whether a lot of these organelle remodeling complexes, a lot of these organelle remodeling events that I told you about are underlying some of these are based, on changes in membrane contact sites, and whether these proteins that form membrane contact sites are facilitating, are driving, or are regulated by these organelle remodeling events. So looking at this during infection, we had to first develop a method that allowed us to detect these hard-to-detect membrane contact site proteins. These are, some of them low abundant, they are membrane bound, we didn't have antibodies for a lot of these. So Caitlin in my lab developed this targeted mass spectrometry assay that allowed her to detect all the known membrane contact site proteins, detect them and quantify them, and then apply this to the 4 different viruses that I mentioned earlier, these DNA viruses, HCMV and HSV1, influenza, and this beta coronavirus, and look at how these contacts are changing as the infection progresses. And this kind of initiated some really interesting research directions in our lab, and I want to tell you what we have learned from her findings on altered mitochondria-ER contacts, especially during HCMV. So if you think about mitochondria, we know that mitochondria are such a hub, regulatory hub during viral infection. So many viruses are targeting mitochondria, either inducing fusion or fragmentation or fission. And this is in order to control respiration, so energy production, immune signaling, or apoptosis. And usually what we know is that when mitochondria are being fragmented, that's connected to a decrease in respiration. That's connected to decreased bioenergetic output. And here you see some viruses that either induce fusion or fragmentation. But something that has been a puzzle in the field of HCMV biology is that this virus, human cytomegalovirus, induces really striking fragmentation of mitochondria, as you see here. But this mitochondria is also bioenergetically enhanced. So we see increased in flux through glycolysis and increased oxidative phosphorylation during HCMV production. So Caitlin discovered that at the time when this increase in oxidative phosphorylation occurs, there is also increased contact between mitochondria and the ER through those membrane contact site proteins that facilitate that contact. So she therefore investigated more this contact, turning to microscopy. And what she found is that this contact is really changed during infection. So in uninfected cells, and these are primary human fibroblasts, she noticed the usual and known type of interaction between mitochondria and the ER, where there is this dynamic interaction. You see mitochondria in red, and ER kind of weaving around the mitochondria and having this dynamic interaction. But after infection with HCMV, and as the infection progressed, the fragmented mitochondria became encapsulated and stabilized in ER pockets. So she termed this new sort of contact structure MENC for mitochondrial-ER encapsulation. And we went ahead and confirmed this using cryo-electron microscopy, and you see here how the fragmented mitochondria is surrounded by the ER. And then we investigated its function. We found what proteins are necessary for inducing the formation of MENC, and we showed that somehow this structure functions for the benefit of the virus. And since then, we investigated how this happens and why, and this was work done by Will Hofstadter, a PhD student in my lab. In other words, how are these MENCs formed, and why are they functioning for the benefit of the virus? And what Will was able to show is, first, looking at how MENCs are forming, he considered two things. He considered how mitochondria are fragmented and how during infection. And he found that HCMV suppresses fusion of mitochondria, but promotes fragmentation through this peripheral fission. So instead of the classic mid-zone fission, it's this kind of newer, newly discovered mode of mitochondrial fragmentation through peripheral fission occurring in the outer part of the mitochondria. And what is known in uninfected cells from Manley's lab is that when mitochondria undergo peripheral fission, this progeny here is actually subjected to mitophagy. So it's induced targeted for degradation, and it also has decreased in membrane potential. But I just told you that during infection, the opposite happens. So what Will showed is that these fragmented mitochondria, all of this, by tracking it, they become encapsulated in this structure in MENCs, and they become protected. So he saw a decrease in mitophagy and demonstrated how this is dependent on the formation of MENCs. And also, he showed that this is connected to increase in membrane potential. So MENCs protect mitochondria, fragmented mitochondria, against mitophagy, and also promote respiration. Downstream of this MENC formation, mitochondria start to communicate with each other, forming mitochondrial-mitochondrial contacts, or IMCs, intermitochondrial contacts, through this ER, and you can see them here, and then they further communicate by starting to swap membrane potential. You can see that here. So this mitochondrial-ER encapsulation that we called MENCs, they protect and maintain the health of fragmented mitochondria in a population. So then we, of course, wanted to ask whether this structure could have some sort of broader relevance. Did we just stumble upon this during HCMV infection? Is it more broadly relevant? And when we think about HCMV, we have to think about different cell types, because HCMV is known to infect many different types of, many different cells. And of course, depending on what virus strains you're using in the lab for studying this, they either have lost or still retain the ability of infecting different types of cells. So we'll kind of tackle these questions using different HCMV strain to infect either fibroblast or epithelial cells and look at their changes and changes in membrane contact sites. And using targeted mass spectrometry assay as well as data-independent acquisition on a new instrument that we acquired, the timsTOF Ultra. So what he found was really interesting, because he found that even for the changes, the membrane contact sites, alteration in membrane contact site abundances that were different in epithelial cells or fibroblasts, there seemed to be some convergence there. So let me tell you what I mean. So if we look here at these are different membrane contact sites that facilitate different contacts. But you can see how their abundance started at a different point in uninfected cell in either fibroblast or epithelial cells. But then as the infection progressed in these different cells, they started to become more similar to each other. And the same here, right? It started as being high in fibroblasts and low in epithelial cells, but as the infection progressed, even if they had different trends, they kind of ended up with a similar protein abundance in these cells. So what this tells us is even if the viruses infect fibroblasts and epithelial cells, there are some convergent changes that are almost pushing this cell towards some sort of unified cell state that is amenable and supports well viral infection. And then there were also shared changes that were the same for any cell that we looked at, and these also included the formation of MENCs. So as I said earlier, we already knew that what membrane contact sites are needed for forming that structure, the MENC, and it's PTP-IP51 on the mitochondrial side and VADP on the ER side. And here you see how this protein changes similarly in abundance as the infection progresses in either fibroblast or epithelial cells. And it similarly recruited in an asymmetric manner at this MENC structure. And we saw this in fibroblasts, epithelial cells, as well as in these macrophage-like cells, so differentiated THP1s. So now we understand that, okay, there is some broad relevance that the formation of MENCs is really broadly important for HCMV infection in different types of cells. Now, the other question was, well, how about more broader context, in other contexts where we do see mitochondrial fragmentation, but in conjunction with this high bioenergetics? And immediately that made us think of cancer, because HCMV is also an oncomodulatory virus. And indeed, Will showed that MENCs are forming in melanoma cells, and Crystal in my lab looked at high-grade serous tubal ovarian cancer and found the formation of MENCs. And furthermore, by looking at different model systems of ovarian cancer, of tubal ovarian cancer, and looking at those associated with poor prognosis, she found that these are exhibiting MENC formation in conjunction with increased oxidative phosphorylation, decrease in immune factors, and increased susceptibility to HCMV infection, again suggesting that MENCs are beneficial for viral infection and promoting or making changes in these cells that are make these cells amenable to viral infection. Okay, so what I told you so far is how our studies of organelle remodeling and organelle-organelle contacts have allowed us to answer this sort of a paradox in the field of HCMV infection, the fact that how fragmented mitochondria still have increased oxidative phosphorylation, and it's through this formation of a mitochondrial-ER encapsulation that protects mitochondria from mitophagy and increases respiration. But another output of these metabolic changes during HCMV infection, and in particular for this increase in flux through glycolysis, is actually an increase in lactate production. And I want to spend part of my talk telling you how we now started to understand how lactate is really important both at the in intracellular and intercellular communication, and placing these in the context of virus microenvironment. So we know that a number of viruses are inducing metabolic changes that mirror what happens in cancer cells. In particular, they induce a Warburg-like effect, where there is a change that's aerobic glycolysis that results in the increase in lactate production. And there haven't been a lot of study of what that means in the context of infection, what's the effect of this increased lactate on infected cells. But outside the context of infection, there have been studies in cancer research that has recently shown that lactate can also inhibit immune responses. So we wondered whether HCMV infection also leverages this increase here in lactate for immune suppression. And in order to address this, Matt, a senior PhD student in my lab, first did a simple experiment where he infected cells with a low MOI so that he can look at spread of infection and induction of immune signaling. And he treated these cells with lactate. And what he found is that in this context of a low MOI, lactate treatment was actually functioning for the benefit of the virus, was pro-viral for subsequent rounds of replication. So what does this mean? Well, from the lab of Inming Zhao at University of Chicago, we now understand that lactate can be added to proteins as a post-translation modification on lysine residues, modifying these proteins by lactylation. So given this sort of increase that we know that HCMV produces in lactate production, we anticipated that probably there are lactylation events happening in the cell. So therefore, we did the lactyl enrichment. This is affinity purification with an antibody against lactylated lysine at different time points of infection with HCMV. And then we use mass spectrometry to start to discover what proteins become modified at these lysine residues by lactylation. And we found that there are a ton of proteins, thousands of proteins in almost every single subcellular compartment that become lactylated during HCMV infection. And then when we started to compare this with other modifications, let's say with acetylation, we found that the motifs were very different, the sites were quite different, and we found that lactylation in particular was enriched in intrinsically disordered regions, and protein intrinsically disordered regions. So then we thought, well, is this just something that we noticed during infection when we compared to other fibroblast data sets and other infected studies during viral infection? Or how about more broadly? So we downloaded lactylation data sets in hepatocellular carcinoma, in E. coli, and again we found that the lactylated modifications resided or were enriched in regions of the proteins that were predicted to be intrinsically disordered. And we used also AlphaFold, and again we confirmed this by seeing an increase in unstructured regions and increase in alpha helix representation in peptides or in regions of the proteins that contain lactyl lysine. So what does this mean? How is lactylation of a protein intrinsically disordered region, how can it impact their function? So we looked at the function class, the classes of proteins that become lactylated as the infection progresses, different hours post-infection, and we found that proteins that become heavily lactylated are involved in immune signaling, so cytokine to response, cellular response to interferon gamma, activation of innate immune responses. And then we asked if this is just the case for HCMV. We performed similar lactylation studies during HSV-1 infection, looking at different viral strains that are either able to inhibit immune responses or unable to inhibit immune responses. And again, we found increased lactylation in proteins involved in immune signaling. So this is a conserved feature of HCMV and HSV-1 infection. So amongst these immune factors that we found to be modified by lactylation was a protein that we care a lot about, that we studied for many years in my lab, which is IFI-16. So this is interferon-inducible protein 16 that we and others have characterized as a viral DNA sensor that can function in the nucleus. And we found that this protein becomes decorated with lactylation in HCMV infection as well as HSV1 infection. And some of these are in these intrinsically disordered regions that we previously characterized. So looking at Matt mutated many of these residues and performed many studies, but I just want to focus on one of these. So looking at one of these sites that he found as lactylated during both infections, he found that when he mutates this site to a lactyl mimic, he actually noticed that this protein starts to have inhibited ability to induce immune signaling. So there are decreases in interferon beta and CXCL10. And in agreement with this, he saw an increase in virus titer when cells were expressing this lactyl mimic protein. Downstream of IFI16, another immune factor, downstream of DNA sensing, RBM14, also here is a lactylation that Matt found in a prion-like domain. And again, when he mutated that residue to a lactyl mimic, he found that this protein starts to have inhibited ability to induce immune signaling, so reduced interferon beta and CXCL10 in conjunction with increased virus production. So to address kind of why that is happening, why lactylation of host immune factors seems to suppress immune signaling, we had to turn back to previous studies in which we tried to put this in broader context and understand how is this DNA sensor even working and what are its interaction needed for inducing immune signaling. And over the years, we have done this in a number of ways. We have used different methods that are available to us to study local protein interactions or global protein interaction. And actually, more recently, we move more and more towards understanding or capturing protein interaction networks at the systems level, because we started to appreciate if we want to understand organelle remodeling and how things are coordinated, we actually need to capture an interaction network more broadly. And for this, we used a method called thermal coaggregation profiling, in which a sample of interest, it can be cells, it can be tissues, this sample is subjected to increasing temperatures. And for each temperature, you would take the remaining soluble fraction and then analyze it by mass spectrometry. So the data would look like this, would be aggregation profile, because as the temperature increases, you have less and less of that protein. It becomes more aggregated, so then you have less in the soluble fraction. But you can look at the similarities of these curves to start to predict interactions, because when proteins are together as a complex, they actually don't behave as separate entities, but rather they become that unit, and they have the properties of that complex unit. So therefore, they have similar aggregation profiles. So we were the first to apply this to viral infection, and we used it to study human cytomegalovirus to find some internalization of receptor. We used it for HSV1, and I'll come back to that. And we also more recently used it to study KSHV, Kaposi sarcoma-associated virus. But the reason why there are very few labs that are using this technique is because these aggregation profiles are really a mess to study. They are really complex. It's very hard to decipher kind of what does this mean in terms of protein interactions. And you need really good computational skills and power to analyze this. So Tavis Reed, a PhD student in my lab, who just recently graduated, he developed a computational platform that he called Tapioca. And this is funny because the name came from someone who kept on not understanding what TPCA means and said tapioca and said, okay, we'll call it Tapioca. So in which he designed this to integrate, so to use logistic regression submodels to integrate subsets of these, what we call dynamic data from TPCA with different static data from tissue-specific functional networks, domains of the proteins, and predicted physical properties of the proteins. And he showed how, using this, you can deconvolute this very complex data, and it doesn't even matter how well behaved or poorly behaved the protein is, but you can use this to really predict de novo interactions. And coming back, why that mattered to our IFI16 story and lactylation story, well, we used TPCA to understand also IFI16 interaction, and this is what it allowed us to further characterize it. So to summarize a little bit, we and others have shown that IFI16 can sense nuclear viral DNA, and it does that by localizing at the nuclear periphery here. As the capsid docks at the nuclear pore and inserts the viral DNA here, IFI16 localizes here. In kind of more recent years, we showed how IFI16, when it's at the nuclear periphery, recruits the DNA damage kinase, DNA-PK, which in turn phosphorylates IFI16 in this intrinsically disordered region that we just talked about. And that phosphorylation we showed to be necessary for IFI16 ability to induce immune signaling. So that it adds to this growing understanding of a crosstalk between DNA sensing and DNA damage response in immune signaling. So we already knew that this intrinsically disordered region is so important for the ability of IFI16 to induce immune signaling, and that it's decorated with this phosphorylation site as well as additional phosphorylation site. And Dawei in my lab more recently has shown that those numerous phosphorylations in the intrinsically disordered region can explain something that we noticed for many years for this protein. That once it binds to viral DNA, it has this really strikingly dynamic behavior, that it starts to form all these puncta, and we never could understand what that quite means. And Dawei showed that IFI16, through these phosphorylations that happen on the intrinsically disordered region, undergoes liquid-liquid phase separation, so this very dynamic behavior, and how this is actually important for inducing immune signaling. So we know that that region, that intrinsically disordered region, is really important and is regulated by post-translation modifications. And now suddenly we know that it's also lactylated, and somehow that lactylation can suppress IFI16 immune signaling. So therefore we went back to our TPCA data set to find out what interactions are affected by that lactylation site. And in looking at this, we found that it actually, when we use the lactyl mimic, IFI16 starts to has impeded ability to recruit the DNA-PK at the nuclear periphery. And as I told you, the DNA-PK is needed for phosphorylating IFI16. So now we understand that lactate is proviral for subsequent rounds of replication. And in part, this is through the induction of lactylation on host immune factors and suppression of cytokine expression. And we started to look at what enzymes are regulating this, and that's kind of another story. But I want to spend kind of a bit of time, the remaining part of the time, to start to put these findings in the broader picture. Because what I told you about lactate, it really brought a new perspective to our long-term interest in understanding how all these changes that I'm telling you about in this infected cell, how are they conveyed to the environment of this cell, to the cells that are in the immediate proximity or those that are further away? How is this infected cell changing its environment? And the lactate, of course, plays a role in this. So a few years ago, we tried to address this, to start to design a method for separating the cells based on how close or far they are from an infection site. To really start to tackle this question of the virus microenvironment, to separate the cells and start to characterize them. And in order to do this, we adapted a method that was previously used for studying the tumor microenvironment. So what we did was to generate fibroblasts that expressed an M-cherry protein, a secretory signal peptide, and lipid-permeable peptide, so making these red cells, which we then infected with one of our green fluorescent virus, HCMV, creating these dually fluorescent cells, red and green. And then we carefully optimized the co-culture of these cells with wild-type cells in order to allow for the spread of this M-cherry signal peptide prior to the spread of an infection. So in other words, we will have the infected cells that are green and red, the neighboring cells that are red, and those that are further away that are not labeled. And then we can use fluorescence-activated cell sorting to sort this cell population, and then use proteomics, use mass spectrometry to characterize their composition, but also do functional studies on the separated cell populations. So what we discovered was that a few really interesting things. So in the infected cell, as we expected, HCMV has mechanisms for suppressing immune signaling. So we found that immune factors are decreased. However, we found that the neighboring cells also had dampened immune signaling, and that the distal cells were the ones that actually had enhanced immune factors. The membrane contact sites that I told you about earlier that represent all those organelle-organelle contacts, we found that they were remodeled in infected cells, But we also found that a specific subset of these membrane contact sites were also changed in neighboring cells, and then they were normal in distal. And finally, probably the most puzzling one is that, yes, we know that viruses target the cell cycle and that they arrest, HCMV, for example, arrests cells at this G1S interface in order to ensure its nuclear DNA replication. But we found that the neighboring cells get arrested in mitosis, in M phase. So now with this sort of lactate story that I told you brought a completely new perspective to this. Now we understand that lactate can be added to immune signaling factors, and that can also, the fact that it's secreted and produced in such large amounts can really dampen the immune responses. So probably it contributes to this effect that we see here. But we also wondered, well, what about all these changes in neighboring cells that seem to be somehow primed with all these changes? What is the relevance of these changes to co-infections or to disease progression? And the reason we are interested in co-infection is, as I said, HCMV infects so many different cell types and so many tissues, and it's known that those can be infected with many other viruses. And there is a lot of interest, of course, in the link between co-infections and morbidity, decreased immunity, cancer development, and so on. So what we did was to generate, again, our virus microenvironment and then test the susceptibility of these cells to infection. So this is a virus microenvironment for HCMV. So these are infected with HCMV. And what we found is that cells neighboring a site of HCMV infection are more susceptible to infection not only with HCMV, but also with HSV1 and also with influenza A, so another nuclear replicating virus, but an RNA virus. We initially did this looking at fibroblasts surrounded by fibroblasts, or at least the cell's background is kept consistent. But then we started to also look at cells surrounded, different interactions between fibroblasts and epithelial cells. And we find again that epithelial cells surrounded fibroblasts are more susceptible to infection with viruses, such as, for example, with HSV1, as I'm showing here. So clearly, viral infection reshapes the microenvironment to facilitate the progression of infection. And we start to have evidence that some of these changes may be linked to oncomodulatory properties, because we have signs that there may be genomic instability in these cells, and also the cell cycle disturbance. So therefore, kind of going forward, we started now to adopt this to look at cell-to-cell communication as it would occur in a tissue, so looking also at direct cell-to-cell contacts. So Crystal in my lab has started developing this method for looking at virus microenvironment in organoids. And I'm showing you just one example here. This is in patient-derived colorectal cancer organoid, where you can see how she can monitor different stages of an infection in this organoid as it progresses. And then she went ahead to separate these cells to characterize them. So since I want to leave a lot of time for questions, I'm going to finish to say that I hope I showed you how we can integrate proteomic and virology studies and microscopy studies to start to look at cellular communication during infection, and that we looked at cellular communication either at the intracellular or intercellular level. So intracellular level, we looked at organelle-organelle contacts that are facilitated or facilitating organelle remodeling, and how this led to the discovery of these mitochondrial-ER encapsulations that we called MENCs, and that protect fragmented mitochondria from degradation by mitophagy, maintaining their increased bioenergetic and increased oxidative phosphorylation. I showed you how those changes that happen in metabolism are inducing also increased lactate production, and why this is important at this interface between disruption of metabolism and immune signaling, because this increased lactate production drives lactylation of host immune factors in their intrinsically disordered regions, and this inhibits their ability to induce cytokine production. And how we learn what specific protein interactions are disrupted using these thermal coaggregation profiling and interaction networks. Finally, I showed you how we started to look at how this information is conveyed to the environment and what's the impact of an infected cell on its neighborhood, and how we found that neighboring cells, so cells neighboring an infected site with cells infected with HCMV, become more susceptible to infection with HSV1 and HCMV and even influenza A. And we are performing quite a few studies understanding those mechanisms now. So with that, I want to thank my lab for all their tremendous work, the funding, including funding from the NIH, from NIID and NIGMS. I recently became, as you heard last year, I became the editor-in-chief of Molecular and Cellular Proteomics. So I want to extend personal invitation for you to submit your work to MCP and invite you to a summer conference that will be in Boston. And I just want to finish by saying that also very recently we lost another phenomenal virologist, Dr. Thomas Christie, who I had the privilege to get the opportunity to work with and collaborate and to call my friend, and he would be absolutely dearly missed. And I just want to send my thoughts and my appreciation for everything that he has done for this community. So thank you so much.
Well, we do have mics, plenty of time for questions. I'll start with one online as we're. Can you attribute the different virus effects to the levels of types of interferons induced?
Different virus effects to types of interferons. Sorry, just to clarify about which one is the question, what it refers to. So,
I see. It came in relatively early in your talk.
Early.
And it's from an immunologist.
Yes. So the type of viruses to say again.
Can you attribute the different virus effects to the levels of types of interferons induced?
Okay. So I'm not sure. So if it's in the first part, it's probably about organelle remodeling and how those different changes in organelle remodeling can also impact immune signaling. I'm going to speculate and answer this in many different ways because I can answer it for different parts.
This person I know, maybe he'll be writing back to clarify.
Yes, great. Okay. So I can also just maybe say something about immune signaling and organelle remodeling because that's something that we were thinking even, let's say, if it's from the first part on mitochondrial remodeling. We were looking at these mitochondria-ER contacts, and as you know, a resident of ER is important protein sting that is important for immune signaling. So we were thinking, okay, these MENG formations are antiviral, but if we induce this contact between mitochondria and the ER early on, it's not antiviral, it's actually proviral because it activates sting. So if mitochondria is not yet fragmented, it's filamentous in nature, and you're tethering it artificially to the ER, it activates sting. So that's, if that question is about whether organelle remodeling and mitochondrial fragmentation is also to suppress immune signaling, it's a possibility. It's also possible that there's just something to do with motility and so on. But my guess is that the clarification will come pretty soon. Yes.
Fantastic talk. I've always loved your growing story on looking at infected versus neighboring versus distant cells is really fascinating. So with the neighboring cells, you also see the MENG changes. And so is that, you touched on this a little bit maybe, but is that priming the cell for virus uptake, for metabolism, dampening the immune response? Like how different is that uninfected neighboring cell from an infected cell?
So it's quite different, and there seem to be only a subset of those membrane contact sites that are changed. And some of them seem to be with regulating calcium transfer. So we see only specific pools of membrane contact sites that are changed in infected cells, in uninfected cells that are neighboring uninfected cells. They look quite different. So I think it's something to do, exactly as you say, with priming those neighboring cells for infection. So that's why we have other projects in the lab where we are looking now at how these are contributing to changes in cellular state. Because we know, of course, viruses can induce mesenchymal to epithelial transition, and something that we were studying in the context of HCMV infection, but or the opposite, EMT. But how do they change also the state of these neighboring cells and furthermore? And that, as you can imagine, can be quite has implications in pathologies. Yeah.
Hi, Eliana.
Hi.
With regards to the MINC part of the story, two questions. One, when they fizz, when the
Yes.
25% fission on the ends, do you see loss of mitochondrial potential before the fission, or is it maintain, or is the MINC just helping to maintain it instead of restore mitochondrial potential? And also, what's the benefit for the virus in making small but highly bioenergetic mitochondria versus the hyperfused type
Yes.
of mitochondria?
Okay, beautiful question. Oh, very beautiful question. So yes, we do actually see a slight decrease in this, and then we see the increase, the rescue, right? In terms of, and it's interesting because we've been monitoring this and trying to even look at temporality and even for that swapping of membrane potential. So it's really interesting how it goes away and comes back. But in terms of the rationale for this, I think there are multiple possibilities. So of course, some of this is known to be connected to regulation of apoptosis, but other can be also with motility and where these mitochondria are being localized. Because we start to see them, how later in infection they go around the assembly complex where, a lot of that rearrangement of the Golgi and endosomes happens, all that organization and production of new infectious viral particles. So maybe energy is brought at the site where it's needed. It's very possible that that's one likely scenario to be demonstrated. Yeah.
I had another MINC question.
Yes.
So you talked about the formation of these structures in HCMV infection and in cancer. Are there non-disease states in which these structures are formed, for example, maybe during development or some other scenario?
Yeah, so really good point, and I think it's connected to the earlier question as well, and so I'm glad you asked it, because the reason why we also started to look in those cancer model systems is because we saw MANCs being formed at a very small level in uninfected cells. So we knew that they can be formed. They are just the infection promotes this and pushes all the mitochondria into this particular state, but they can exist. So that's why we thought, okay, they exist also in uninfected cells, and then now we know that they are even connected to more like poor prognosis. And I would say that yes. So in terms of your last question, we started to look at some of the neurodegenerative diseases like Alzheimer's and Huntington's and so on, and perhaps in Alzheimer's. So it's possible. It's possible that maybe this is a mechanism to kind of protect fragmented mitochondria that still needs to produce a lot of oxidative phosphorylation, produce energy. It's possible. It just remains to be seen. We started to look into that a little bit, but just very recently. Yeah. One thing that I will mention is that, and kind of connected to, actually I'll answer your question first. Yes.
Well, I have three, so
yeah, perfect. Then you may
if you can come back
you
to
may
it.
ask exactly what I was going to say.
Two technical questions to start with. For the formation of MANCs, have you identified any RNA markers where people can look at the RNA level to see if that suggests increased formation of MANCs?
No, so we have not.
Okay.
No, but the detection of the MCSs themselves, the ones that we found to be localized there, is really, they provide a great marker, because this is one of those few examples where a change in the abundance of a protein actually really signifies a change in the extent of contact between two organelles. So, because usually, you find changes in abundance, and what does that mean? It can mean a lot of different things. But there probably are also other markers that are not at the RNA level, but rather at the protein level. I would not be surprised if some, let's say, post-translation modifications are important for targeting. It's known that that happens for certain MCSs, that they need to be modified in certain ways in order to be recruited to a certain contact for proteins that have multiple localizations. Yeah.
Okay. And are there antibodies that are now specific to lactylation sites in proteins? Are these like reagents that people can think about using?
There are antibodies that are specific to lactylated lysine.
Okay.
Not necessarily to a site in a protein. And the initial studies on lactylation the initial studies have been done on histone modifications and how, nuclear lactylation is important and why this is important just in terms of possible even crosstalk with other PTMs on histones and gene expression. So this is kind of how that field or post-translation modification of lactylation has started. Now we understand that lactylation can really be broadly regulated, and we have started to look at what enzymes in the context of infection are regulating this modification, and it's really fascinating. We found that an enzyme that is involved in placing alanine, so on tRNA, so alanine tRNA transferase is the one that brings the lactate on proteins. So you can push this enzyme. If you feed cells alanine, you're making it into an alanine tRNA transferase. And if you feed them lactate, it becomes, so it's called AARS1, that it functions and works in the nucleus as well and in the cytoplasm, and ARS2 that functions in the mitochondria for some of the lactylations that we've seen.
I see. Okay, one more quick question. In the last part of your talk, you mentioned that the neighboring cells had a dampened sort of immune response state, but the distal ones had an enhanced state. Do you have ideas of the signaling that's happening between the neighboring cells and the distal cells?
No, but we started to look at this, and also from the perspective of heterogeneity of responses. So within each of these populations, as you can imagine, let's say the neighboring, the distal, there's still such heterogeneity of response to viral infection and different susceptibility still to infections. So we started now to separate these, and a bit more than a year ago, we installed an instrument in the lab that can do single-cell proteomics. So now we are separating all these cells and are looking at the single-cell level, kind of what composition represents a certain response or ability. And we are getting also to that question of interactions between the neighboring and the distal cells.
Name.
Yeah.
Thanks.
Another lactylation question. You mentioned that lactylation prevents the recruitment of kinases. Do you know the mechanism for that? Is it simply steric hindrance, or is there something more subtle going on?
Yeah, so that intrinsically disordered region that we find to be modified, so we find that many times what happens to that region has a lot to do with charge interactions. And maybe that's what it's about, and how it suddenly folds in, creates a different fold, a different environment. So we do not know more. All we know for the moment is that when the lactylation is present on the intrinsically disordered region, suddenly DNA PK can no longer be recruited to the nuclear periphery at sites of genome deposition. That's kind of where we are with that for the moment.
Any effect on protein stability?
No, we did not see an effect on protein stability. But I want to also mention something else on lactylation, because just for the flow of the talk and interest of time, I wanted to give you only the story about the immune signaling and the lactylation on immune factors. But we found, obviously, a lot of proteins being lactylated, right? So it's a little bit like phosphorylation. You can't say that something is antiviral, proviral, because you're going to have thousands of proteins, and they will have so many functions. So what you're looking at is really an averaging of that. But we also found viral proteins that become lactylated. And some of these viral proteins, when we did the same experiment I showed you earlier with lactate treatment and low MOI, but when we did the same experiment with high MOI, we found that lactate was actually antiviral. And so in the infected cell, when all the cells are infected and we are not looking at viral cell-to-cell spread and immune signaling, we found that then a lot of viral proteins become lactylated. Some of these intrinsically disordered regions become lactylated. And one of them had to do with, it was, again, inhibiting the ability of that IDR, of the viral protein, to go into a liquid-liquid phase separation in the nucleus to go into replication compartments. So it can also inhibit the function of a viral protein. So it has lots of, lactylation can alter protein function in many different ways.
Okay. Do we get a clarification on the?
I think he was, that individual was all set with that response. So,
Oh,
great.
okay.
Thank you.
Any other online questions?
No, that's it. And if you want to wrap up, oh, let me say the CME code. Oh, there it is, 57688. And there's a
All right.
reception you can mention. And
Yeah, there's a reception outside immediately afterwards, and let's thank Ileana for a really beautiful talk.
Thank you so much.